So let's start with glycolysis. So glycolysis is the common pathway of both aerobic and anaerobic respiration, and this glycolysis is also called as EMP pathway. EMP pathway, because the details of this glycolysis is given by three scientists: Empeden, Meyerhoff, and Parnas. Hence it is also called as EMP pathway. So this glycolysis involves 10 steps. It is a 10-step pathway. 10-step pathway, and these 10 steps are divided into two phases. They are first phase and second phase of glycolysis. Second phase. So first 5 steps are included under first phase of this pathway. 1 to 5 steps are included under first phase, and whereas 6th to 10th step, 6 to 10 steps are included under second phase of this pathway. Okay, so this first phase of this pathway, what happens during this first phase? Energy is consumed. Hence this phase is also called as energy consuming phase. Energy consuming phase, or it is also called as glucose activation phase. Glucose energy consuming, or we can say energy spending phase. Spending phase. And it is also called as glucose activation phase, and it is also called as preparatory phase. Preparatory phase. And second phase is called as energy payoff phase, because in the first phase energy is consumed, in the second phase energy is released. Energy is released. Hence it is called as energy releasing phase or payoff phase, or also called as energy conserving phase. Conserving, not consuming, conserving phase. Conserving phase. Okay, now let's start with all these details of glycolysis, 10 steps which are there in glycolysis. Okay, let's see the details of these 10 steps of glycolysis. Okay, so glucose, which is a 6-carbon compound, undergoes phosphorylation to form glucose 6-phosphate. Phosphorylation means addition of phosphate. So this phosphate, where it is added at the 6th carbon atom of the glucose, and hence glucose has become glucose 6-phosphate. And this phosphate is donated by ATP, adenosine triphosphate. After donating one phosphate, it has become ADP, adenosine diphosphate. And this reaction, first step of this glycolysis, is catalyzed by the enzyme hexokinase. And for the activation of this hexokinase, Mg+2 magnesium ions are required. Next, in the second step, now this glucose 6-phosphate undergoes isomerization into fructose 6-phosphate. Isomerization, both of this glucose 6-phosphate and fructose 6-phosphate are interconvertible isomers, and this isomerization is catalyzed by the enzyme phosphohexose isomerase, and for its activation also magnesium is required. Next, in the third step of glycolysis, this fructose 6-phosphate undergoes phosphorylation. Phosphorylation, again addition of phosphate. Phosphorylation into fructose 1,6-bisphosphate. So here also phosphate is donated by ATP, and after donating phosphate, it has been converted into ADP, adenosine diphosphate. And this step is catalyzed by an enzyme phosphofructokinase. Phosphofructokinase is an important enzyme of glycolysis, which is involved in controlling or regulating the process, the pathway of glycolysis. Hence this phosphofructokinase is also called as pacemaker enzyme of glycolysis. Pacemaker enzyme of glycolysis. So fructose 1,6-bisphosphate or biphosphate is formed. So where this phosphate is added, new phosphate, it is added at the first carbon atom of the fructose. Already phosphate is present at 6th carbon, 6th position. So hence it has become fructose 1,6 bisphosphate or biphosphate. Two phosphates are present, biphosphate, which is 6 carbon compound. Now in the fourth step, this fructose 1,6 bisphosphate undergoes cleavage. Cleavage into two molecules of 3 carbon compounds. Two different molecules of 3 carbon compounds. So this cleavage of this fructose 1,6 bisphosphate is catalyzed by an enzyme aldolase. So it is cleaved into one molecule of 3 carbon compound called DHAP, dihydroxyacetone phosphate, and another molecule of 3 carbon compound, that is 3-phosphoglyceraldehyde, which can also be called as glyceraldehyde 3-phosphate, which is 3 carbon compound. Now both of these compounds are interconvertible isomers, and hence what happens now in the fifth step, DHAP undergoes isomerization into G3P or 3-PGAL. So this isomerization is catalyzed by the enzyme triose phosphate isomerase, right? So what happened now, by the end of this first phase, so we already discussed it. Glycolysis involves two phases, first and second phases. First phase is nothing but first to fifth step are under first phase, that is energy consuming phase or activation phase or preparatory phase. So by the end of this fifth step, preparatory phase is completed. Now 6th to 10th step together are called as energy release phase or payoff phase or conservation phase, energy conservation phase, right? So by the end of this step, now two molecules of PGAL is formed or G3P is formed. So from here we are taking two molecules, but till here we are taking only one molecule. Now two molecules of G3P, glyceraldehyde 3-phosphate or 3-phosphoglyceraldehyde, now undergoes oxidative phosphorylation to form two molecules of 1,3-bisphosphoglyceric acid, and this step is catalyzed by an enzyme G3P dehydrogenase. Now let's see what happens during this step, sixth step. So G3P undergoes oxidation or breakdown, which is nothing but dehydrogenation. So when it undergoes oxidation or breakdown, what happens? Two redox equivalents, that is two protons are released. These two protons which are released are transferred onto, they are picked up by NAD+. So this NAD+ is reduced into NADH plus H+. So due to oxidation of G3P, two redox equivalents, that is nothing but protons or H+ ions are released from this, which are picked up by NAD+. So that NAD+ is converted into NADH and H+. So this is called oxidation or dehydrogenation, due to which two molecules of NADH plus H+ is formed. Why two molecules? Because two molecules of G3P undergoes oxidation. Next, phosphorylation, oxidative phosphorylation and now phosphorylation. So phosphorylation is nothing but addition of phosphate. So phosphate is added in the form of H3PO4. In this step, phosphate is added in the form of H3PO4. So here H3PO4 is with phosphate, that is inorganic phosphate, and it is also with water molecule. So two molecules of H3PO4 is added. So due to which one inorganic phosphate is added. So where it is added? At the first position of this G3P. Hence it has become 1,3-bisphosphoglyceric acid. Due to the addition of phosphate, that is phosphorylation, at first position it has become 1,3-bisphosphoglyceraldehyde. Now when it will become acid glyceraldehyde, when water molecule is added, it has become glyceraldehyde. So due to oxidation or dehydrogenation, two molecules of NADH2 is released, NADH plus H+ is released, and due to phosphorylation and hydration in the form of H3PO4, G3P has been converted into 1,3-bisphosphoglyceric acid, and this step is catalyzed by the enzyme G3P dehydrogenase. Okay, now in the seventh step, two molecules of 1,3-bisphosphoglyceric acid undergoes dephosphorylation, that is removal of phosphate group. Dephosphorylation into two molecules of 3-phosphoglyceric acid. So where from this phosphate is removed? It is removed from first position. So it has become 3-phosphoglyceric acid. So when this phosphate is removed, this phosphate is transferred onto ADP, so that ADP has been converted into ATP, so that ATP is formed. This step is also called as SLP, substrate level phosphorylation, or direct formation of ATP. What is this substrate level phosphorylation? Now let's define substrate level phosphorylation. Substrate level phosphorylation means synthesis of ATP from ADP due to the transfer of inorganic phosphate from a phosphorylated intermediate or substrate to ADP for the formation or for the synthesis of ATP. That is called substrate level phosphorylation. So substrate level phosphorylation is the transfer of phosphate, inorganic phosphate, from a phosphorylated intermediate to ADP for the synthesis of ATP is called as substrate level phosphorylation, which is nothing but direct formation of ATP. So this is catalyzed by an enzyme phosphoglycerokinase, and again it is activated by magnesium. So this is 7th step. Now in the 8th step, these two molecules, as there are two molecules of ATP is released. So now these two molecules of 3-phosphoglyceric acid undergoes isomerization into two molecules of phosphoglyceric acid. So this isomerization is catalyzed by phosphoglyceromutase. Okay, it is also called as intramolecular shift, because here what happens, phosphate group present at 3rd position has been shifted on to 2nd position. Hence 3-phosphoglyceric acid, 3-PGA, has become 2-PGA. So intramolecular shift, phosphate has been shifted from 3rd position to the 2nd position present inside the same molecule. Hence it is called as intramolecular shift. Right. Next, coming to 9th step. So in this 9th step, two molecules of 2-phosphoglyceric acid undergoes dehydration, that is removal of water. In dehydration into two molecules of phosphoenol pyruvic acid or pyruvate. So this step is catalyzed by enolase. So due to dehydration, what happens? One molecule of water is released from one molecule of phosphoglyceric acid. As there are two molecules of water is released. Next, now these two molecules of phosphoenol pyruvic acid in the 10th step undergoes dephosphorylation. Again it is SLP. Dephosphorylation, 10th step, to form pyruvic acid. So as dephosphorylation occurs, phosphate which is released from this intermediate has been transferred on to ADP for the synthesis of ATP. Hence it is also called as SLP, substrate level phosphorylation, or direct formation of ATP. So this is all about the 10 steps of glycolysis. So by the end of glycolysis, what happens? One molecule of glucose, that is 6-carbon compound, undergoes partial oxidation into two molecules of pyruvic acid by the end of glycolysis. Pyruvic acid is 3-carbon compound. Now what happens to this pyruvic acid? This pyruvic acid enters into either aerobic respiration if oxygen is available inside the cell, or it enters into anaerobic respiration, that is fermentation, if oxygen is not available inside the cell. Okay, so here I would like to discuss some questions related to this glycolysis. Some questions. Okay, so in which step or at what steps this isomerization occurs? So isomerization occurs at 3 steps. They are 2nd step, and then 5th step and 8th step. So isomerization occurs at 3 steps in glycolysis, that is at 2nd, 5th and 8th step. Next, coming to substrate level phosphorylation. At how many steps substrate level phosphorylation occurs? Substrate level phosphorylation occurs at 2 steps, that is at 7th step and 10th step. That is substrate level phosphorylation. At how many steps phosphorylation occurs? Substrate level phosphorylation is dephosphorylation, removal of phosphate, whereas phosphorylation is addition of phosphate. So at how many steps phosphorylation occurs? It occurs at 3 steps, that is at 1st step, and then 3rd step, and then 6th step. 6th step is oxidative phosphorylation. In this, phosphorylation is also included. So at 6th step also, that is addition of phosphate. Now at how many steps oxidation or dehydrogenation occurs? It occurs at only 1 step. How many steps? One step. That is at sixth step. Oxidation or dehydrogenation, where hydrogens are removed. Dehydrogenation. Dehydration is different from dehydrogenation. Dehydration is removal of water. Dehydrogenation is removal of hydrogens. Oxidation is nothing but dehydrogenation. Okay, next, at how many steps dehydration occurs? Dehydration occurs at one step, that is at ninth step. Now, at how many steps hydration occurs? Hydration occurs at one step, that is at sixth step. So, here in the form of H3PO4, water molecules are utilized. So, hydration occurs at one step, that is at sixth step. Next, at how many steps decarboxylation occurs? There is no decarboxylation, that is, there is no release of carbon dioxide during this glycolysis. And at how many steps carboxylation occurs? Carboxylation is addition of carbon dioxide, decarboxylation is removal of carbon dioxide. So during glycolysis there is no addition of carbon dioxide, carboxylation, and there is no removal of carbon dioxide, that means decarboxylation. So this is all about glycolysis. In the next video, I will be explaining about the output of glycolysis, that is balance sheet of glycolysis. Hope you understood. Thank you, students.